Good morning. This is Dr.R.Kamalambigeswari, Associate Professor from Department of Industrial Biotechnology. And today I'm going to present on the topic replication in prokaryotes. This is a central dogma of life. The central dogma of life comprises three different processes like replication, transcription, and translation. The replication is a process where the DNA is going to make the exact copy of its own. And the transcription is a process where the RNA is going to be transcribed from DNA. And translation is a process where the protein is going to be synthesized from the transcribed RNA. And we have one more process called reverse transcription that occurs in many of the viruses, like the RNA will be converted to DNA. And coming to DNA replication, it is a biological process that occurs in all living organisms where the DNA is going to make an exact copy of its own. And once the cell need to replicate, or once a cell want to divide, then of course the DNA replication proceeds before the cell division. And it is the basis for biological inheritance. And the replication is a process of synthesis of daughter DNA from parental DNA, which is mediated by the enzyme Dna Polymerase. And here, in case of DNA replication, we have a parent strand, and the parent strand is going to make an exact copy of its own, like the new strand that is synthesized is said to be the daughter strand. And coming to DNA replication, it's a reaction in which the daughter DNA is going to be synthesized using the parent DNA as the template. The strand which is going to serve as the template for the synthesis of the new daughter strand will be the parent genome. And here, in case of DNA replication, the transfer of genetic information to the descendant generation occurs with a high fidelity, with a high confidence. And here we have the parent genome, which is going to be replicated into two daughter genome. And coming to the patterns of replication, there are three possible replication patterns. One is semi-conservative mode of replication. The next one is conservative mode of replication. And the third one is dispersive method of replication. And among them, the semi-conservative method of replication is accepted unanimously. And this is the one which occurs in the biological organism. Coming to the semi-conservative method of replication, we have the parent genome, and after one round of replication, the new DNA, the new genome that is going to be segregated to the daughter cells will consist of one parent genome and one daughter genome. And in case of conservative method of replication, the genome will be segregated in such a way that one daughter cell will consist of only parent genome and the other daughter cell will consist of only daughter genome. That means the newly synthesized genome. And the third method of replication model is dispersive method of replication. And here there is no proper segregation of parent genome and the daughter genome. It is highly dispersed. The daughter genome and the parent genome are highly dispersed and segregated to daughter cells. This is what is explained in dispersive method of replication, but the semi-conservative method of replication is unanimously accepted, and it is explained and it is proved that it exists in all the organisms, living organisms. So this is how the daughter genome is going to be synthesized from the parent genome. We have the original template strand, and the DNA polymerase will be helping in the synthesis of daughter genome from the template strand. Coming to the direction of DNA replication, generally it was presumed earlier to have two different models like unidirectional method of replication and bidirectional method of replication. Earlier it was proved that the bidirectional replication is going to happen in all the cells. So in case of bidirectional replication, the origin of replication will be at the center of the genome and the replication fork moves towards both the directions, say in clockwise direction as well as in counterclockwise direction. In case of unidirectional replication, the origin is presumed to be at one site, and the moment of replication is found to be at only one direction. But later it was restored, and it was proved like the bidirectional replication was followed in all the organisms. Coming to the bidirectional replication, the replication starts from unwinding the double-stranded DNA at a particular point, followed by the synthesis of each strand. And the parental double-stranded DNA and two newly formed double-stranded DNA form a Y-shaped structure, which is called as replication fork. So this is the movement of replication fork. Here we have two strands like leading strand and lagging strand. And in case of leading strand, the replication is continuous. And in case of lagging strand, the replication is discontinuous. So here the replication fork moves in both the directions. Coming to the replication of prokaryotes, the replication process starts from the origin and proceeds in two opposite directions. And hence it is said to be a bidirectional replication. And the replication is also named to be data replication. Here, if we see the structure of the replication, then of course it is presumed to have a data structure, and hence it is said to be a data replication. Now we'll see about various enzymes and proteins that are involved in replication. So here, the main enzyme that plays a vital role in replication is DNA polymerase. And we have three different types of DNA polymerase as far as prokaryotes are concerned. They are DNA polymerase 1, 2, and 3. And coming to this DNA polymerase, this is the vital enzyme, which is going to play a unique role like polymerization. The DNA polymerase actually matches the correct nucleotide, then joins and polymerizes the adjacent nucleotide to each other. Thereby, it is going to extend the strand. Coming to helicase, this is another important enzyme. It's going to unwind the DNA and melts the DNA. That means the unwinding of double-stranded DNA into two single-stranded DNA is done by this enzyme called helicase enzyme. And this helicase enzyme plays an important role because as the helicase enzyme moves ahead, the replication fork also moves. Thereby, the movement of the helicase enzyme on the double-stranded DNA is highly essential for unwinding the DNA to a single-stranded DNA. Next, another enzyme that is involved in replication is primase. This primase provides an RNA primer to start the polymerization. This for the synthesis of DNA, the RNA primer is essential. The DNA polymerase can add the NTP to the existing primer, but it could not synthesize the RNA primer. So that synthesizing ability is with the enzyme primase, and this primase provides an RNA primer to initiate the DNA replication. Next is about single-stranded binding proteins. So this enzyme is this protein is highly essential since because it is going to keep the DNA single-stranded after it has been melted by helicase. Uh, the tendency of the DNA will be like it just want to wind each other, and it will be existing in double-stranded form. So this single-stranded binding proteins will bind to the single-stranded DNA, and it helps to keep the single-stranded DNA in single-stranded form. And the next enzyme that plays a unique role in DNA replication is gyrase. So this is a topoisomerase enzyme that relieves the torsional stress or strain in the DNA molecule. Since because the DNA has been in single-stranded form during the mode of replication or during the time of replication, there will be high stress to the DNA strand so as to keep the DNA strand in single-stranded form. That stress will be released by the binding of gyrase. So this is an topoisomerase enzyme, and it is going to release the stress or strain in the single-stranded DNA, and it helps to uh, maintain the single-stranded DNA in single-stranded form. And next enzyme is ligase. So this ligase actually is going to seal the nick. So it just joins the DNA strands together, and it fixes the nick. If there is some gap in the DNA strand, so that gap, which is called as nick, will be sealed by this enzyme called ligase, and hence this ligase enzyme is also an important enzyme that plays a role in DNA replication. Next is about telomerase. So this is a unique enzyme, and it finishes off the ends of the DNA strand as far as eukaryotes are concerned. Coming to the DNA polymerase, as I mentioned earlier, there are three different types of DNA polymerases in prokaryotes. So they are DNA polymerase 1, 2, and 3. So the first DNA-dependent DNA polymerase, which is said to be DNA polymerase 1, was discovered in 1958 by Arthur Kornberg, who received Nobel Prize in Physiology and Medicine in the year 1959. So he is a very renowned scientist, and he has received received Nobel Prize also. And so this DNA polymerase enzyme is often called as Kornberg enzyme, just because he was the one who discovered this DNA polymerase. Later, upon years of its discovery, the DNA polymerase 2 and DNA polymerase 3 were also identified. And all of this DNA polymerase enzyme, say 1, 2, and 3, possess the following biological activity, like 5' to 3' polymerase activity and exonuclease activity. The 5' to 3' polymerase activity is the one where the DNTPs can be added to the uh, existing chain or to the growing chain, and the chain length continues. And the exonuclease activity is the one in which if there is any mismatch base, or if the primer needs to be removed, then the exonuclease activity of this DNA polymerase enzyme will be helpful in removing the mismatched base, or it will be helpful in removing the primer, especially the RNA primer. And this is the comparison of DNA polymerases of E. coli. So as we have mentioned earlier, we have three different types of DNA polymerase, like 1, 2, and 3. So coming to the structural gene that is synthesizing this enzyme, the DNA polymerase 1 is synthesized by Pol A, the DNA polymerase 2 is synthesized by Pol B, and the DNA polymerase 3 is synthesized by Pol C gene, respectively. And coming to the subunits, so DNA polymerase enzyme, one enzyme has a single subunit, and polymerase 2 has 7 subunit, and polymerase 3 has 10 subunits. And this is regarding the molecular weight and 3' to 5' exonuclease activity. So all the enzymes has 3' to 5' exonuclease activity, and 5' to 3' exonuclease activity is rendered only by DNA polymerase 3. DNA polymerase 1, whereas DNA polymerase 2 and 3 doesn't have the 5' to 3' exonuclease activity. And the polymerization rate is about 16 to 20 nucleotide per second. That means the ability of enzyme to add the nucleotide in the growing chain is about 16 to 20 nucleotide per second for DNA polymerase 1. And for polymerase 2, it is about 40 nucleotide per second. And for polymerase 3, it is 250 to 1000 nucleotide per second. And coming to the processivity, processivity can be defined as the ability of enzyme to add nucleotide before it dissociates from the DNA chain, from the peptide chain especially. So it is about 3 to 200 nucleotides for DNA polymerase 1, and for polymerase 2 it is about 1500, and for polymerase 3 it is more than 5 lakh. So the polymerase 3, DNA polymerase 3, is said to be the principal enzyme that is involved in DNA replication because of high polymerization rate and because of high processivity of the enzyme. And next is about the exonuclease function. So the 5' to 3' exonuclease activity is necessary for removing the primer, or it is necessary for excising the mutated segment. For example, if any basis has been added due to some mutation, if any wrong base has been added, then of course the 5' to 3' to 3' hexanuclease activity of the enzyme will be helpful in removing it. And 3' to 5' hexanuclease activity will be useful in excising the mismatched nucleotide from the DNA strand. So that is shown here. And coming to the DNA polymerase enzyme, this is mainly responsible for proofreading, and it will be helpful for filling the gaps where and all the nick is there. So the DNA polymerase I enzyme will be helpful for repairing the DNA damage. And proofreading is a process where after addition of nucleotide in the DNA chain, the polymerase checks whether it has added the correct base at the which is complementary to the template. So that activity is rendered by DNA polymerase I, and also it is helpful for filling the gaps, which is called as nick, during repairing the DNA damage. And coming to DNA polymerase II, this is temporarily functional when DNA polymerase I and DNA polymerase III are not functional. So this is still capable for doing synthesis on the damaged template. It is one of the enzymes that is often involved in DNA repair mechanism. And coming to DNA polymerase III, this is one of the important and vital enzyme that takes part actively in DNA replication. It is a heterodimeric enzyme which is composed of 10 different subunits, and this have the highest polymerization activity, about 10 to 5 nucleotide per minute. And this is the true enzyme that is responsible for the process of elongation. Coming to the structure of DNA polymerase III, this is its structure. So it is a heterodimeric enzyme which has more than 10 different subunits. So it has 5' to 3' polymerizing activity. That means it can add the DNTPs to the growing chain in the direction of 5' to 3'. And it has 3' to 5' exonuclease activity, so that we have mentioned in the earlier slide, and it plays a key role to ensure the replication fidelity. So this replication should have to be carried out with a high fidelity, since because the cell will take its utmost care to avoid error in replication. So uh, it need to be a highly fidelity process, and the DNA polymerase III will be helpful to ensure the replication fidelity. And the among them, the theta subunit is helpful for maintaining the heterodimeric structure, and the alpha subunit is responsible for polymerizing activity, and epsilon has 3' to 5' exonuclease activity. And the nucleotides are always added to the growing strand at the 3' end, the end at which the DNA strand has a free OH group on the 3' carbon of its terminal deoxyribose. So this 3' OH will be helpful for adding the nucleotides. So the 3' OH group of the earlier base will be having bond with the 5' phosphate of the incoming nucleotide, thereby the chain length will increase as far as the daughter genome is concerned. And coming to the DNA replication in prokaryotes, it is a bidirectional replication, and it has a single origin, which is said to be the origin of replication, which is mentioned here as origin. And the E. coli replication is circular. Of course, there is no free end. And coming to the replication, it is going to be studied in 3 different steps, like initiation, elongation, and termination. And of course, this is to be the theta replication. I think we have discussed about this in the earlier slide itself. Here, the DNA replication is can be studied in 3 different steps, like initiation, elongation, and termination. Coming to the replication in process in prokaryotes, the DNA replication includes the initiation. The replication begins at origin of replication, and elongation. The new strands of DNA are synthesized by DNA polymerase, and termination. the replication is terminated differently in case of prokaryotes and in case of eukaryotes. Actually, the replication begins during the stage of initiation, and the chain elongation will be there. The chain length will increase at the stage of elongation, where the DNA polymerase plays its vital role. And the termination, the replication is terminated differently in prokaryotes as well as in eukaryotes. So, in case of termination, the replication actually halts. Coming to the process of initiation, the DNA as the enzyme is going to recognize the origin sequence and opens duplex at a specific site. So here we have the 9 base pair repeat where the DNA is going to actually bind and it is going to unbind the DNA strand. And denaturation of DNA and varices requires histone-like HU protein and ATP. The denaturation actually it is going to takes place at the origin of replication, and for that it requires histone protein like HU protein, as well as it is an energy consumption consuming process where ATP is going to be hydrolyzed. And next, the DNA B helicase enzyme, which is a hexamer, binds to the unwound DNA, and the binding of this DNA B helicase will unwind the double-stranded DNA to single-stranded DNA. And binding of DNA B to individual strands also requires DNA C. Uh, the DNA B helicase will in turn need help from another protein, which is said to be DNA C. So DNA gyrase comes into role here for which unwinds the DNA strand, as well as it is going to relieve the topological stress that is rendered by the unwinding of DNA strand. And this is the initiation of DNA replication in detail. Here, the 2 to 4 molecules of DNA bind to the origin of replication, which results in folding of DNA around the aggregate where the proteins have been mounted, and exactly the double-stranded DNA starts to melt from here. And an aggregate of about 6 molecules of DNA B as well as DNA C binds to each of the 3 separate single-stranded regions, which is produced by DNA A. And the aggregate eventually displaces DNA A and DNA C, which is going to load only DNA B helicase, thereby the unwinding of strand progresses. The DNA B function as helicases, and the unwinding begins. And of course, the DNA gyrase will be loading on the single-stranded DNA so as to relieve the topological stress. And the SSB protein will bind to the single-stranded region, and it stabilizes the single-stranded DNA to be in the single-stranded form. And one DNA B hexamer binds to each of this 2 replication fork, thereby the unwinding will be progressed. So once the replication has been started at the origin, and when the primer initiation begins, the primer synthesis begins, which the process is said to be priming. And the priming occurs only once in case of leading strand, and the primers occurs repeatedly. The priming occurs repeatedly at intervals of about 1000 to 2000 bases in case of lagging strand, since because in lagging strand the replication is not continuous, it is a discontinuous replication. And the primosomes moves along the single-stranded region. The primosome is nothing but the complex of enzymes and proteins that is involved in the replication process. And when the primosome reaches a site at which a priming can occur, it is going to synthesize an RNA primer, and the RNA primer is the one which is going to initiate the DNA replication, and the primer sponsors the synthesis of new okazaki fragments. And coming to the elongation of DNA replication, the elongation requires the assistance from several 7 different proteins like SSB protein, single-stranded binding protein, which is going to bind with single-stranded DNA, and DNA B helicase, which is going to unwind the DNA, and primase, where the RNA primer synthesis is going to be initiated by the primase enzyme, and DNA Polymerase III, which is a principal enzyme which is involved in DNA replication, which will be helpful for new strand elongation, since because the polymerization is going to be rendered by this DNA polymerase 3, and the DNA polymerase one enzyme. It is going to remove the RNA primer, and of course, it is going to fill the gap. It is going to seal the nick by synthesizing nucleotides. And the DNA ligase enzyme is going to seal the nick that is caused by uh, uh, due to the um, uh, movement of the DNA strand, and DNA gyrase, which is one of the enzyme that is going to release the topological stress. And here in this representation, we could see the movement of DNA polymerase 3 enzyme. An enzyme holds the replication fork, which is going to add the nucleotides, DNTPs, to the growing chain. And we have a leading strand, and here the lagging strand is also there. And in case of lagging strand, the DNA synthesis occurs in a semi-discontinuous way, in a discontinuous mode, and the short fragments that are synthesized is said to be Okazaki fragments, and these Okazaki fragments will be joined so as to make the chain continuous. And coming to the elongation, the two strands has been uh, take part in elongation, that is leading strand and the lagging strand. And in case of leading strand, the DNA replication is continuous, and then 5' to 3' direction, and the leading strand is the one which moves towards C. In case of leading strand, the DNA strand has been synthesized towards the movement of replication fork. Here we could see the leading strand and lagging strand, and from here itself we could find that the leading strand is continuous. The synthesis of DNA is continuous in leading strand, whereas in lagging strand it is discontinuous. We can see the fragments of DNA getting synthesized in small as a small chain, and this is said to be the Okazaki fragment. The Okazaki fragments will be joined so as to make the daughter genome, so as to make the new strand continuous with the help of, say, DNA ligase and DNA polymerase. And coming to primosome, the primosome complex contains primase as well as the helicase enzyme, and this is responsible for initiation of DNA replication by forming the RNA primer. And primase enzyme, it actually synthesizes the RNA primer, and it provides the free 3' of the chain for DNA polymerase 3 so as to add the nucleotides. Okazaki fragments. So, in case of lagging strand, the DNA synthesis occurs with the help of Okazaki fragments. So, the Okazaki fragments was discovered by Reiji Okazaki, and it is about 1,000 to 2,000 nucleotide sequence, which is separated by 10 nucleotide RNA primer. For the initiation of every Okazaki fragments, the RNA primer is essential. So the red color denotes the newly synthesized strand, and the Okazaki fragments are synthesized as short DNA fragments. And later, these Okazaki fragments can be joined, and the lagging strand will also be made continuous once the elongation is in progress. Lagging strand, it consists of discontinuously elongating strand, and it is away from the replication fork, and the chromosome formation is there in case of lagging strand. And DNA polymerase 1 and DNA ligase enzyme, it is going to uh, uh, replace the RNA primer, and it is going to join the Okazaki fragments. So this is a representation which we have seen earlier. So the lagging strand synthesis is in progress with the help of Okazaki fragments, and the most recently synthesized DNA is in short stretch of DNA segment. Coming to the speed of elongation, due to continuous and discontinuous elongation, leading strand should elongate quickly, and this is prevented by helicase, which acts as a temporary halting signal, stopping the DNA unwinding. Since because in case of lagging strand, the the DNA segment is synthesized discontinuously, and in case of leading strand, since the synthesis is continuous, there may be some the DNA helicase enzyme acts as a temporary halting signal, which is stopping the DNA unwinding, and hence the leading strand won't also be synthesized very quickly. The speed is an equal range in case of leading strand and the lagging strand synthesis. So here we could able to infer the assembly of the primosome and the synthesis of leading strand as well as the synthesis of lagging strand. Coming to the termination of replication, the termination is a process where the DNA replication is going to get halted, and here two replication forks. We have seen the replication fork moves bidirectionally in case of prokaryotic genome, and the replication trap actually it occurs at a specific sequence called a TER sequence. TER stands for termination sequences. So the two replication fork, which is going to move in say clockwise movement, which is going to move in clockwise direction as well as in and counterclockwise direction, is going to be trapped at some region called TER region. So the TER region consists of several proteins like TER A, D, and B, which is in counterclockwise fork trap, and TER G, TER F, TER B, and TER C in case of clockwise fork trap. So once here the TER region is a unique, TER sequences are very unique, that it will allow the unidirectional movement of replication fork. The replication fork can enter the TER region, but it can't leave the TER region. Once the replication fork enters the TER region in a single direction, then of course it is trapped, and hence the replication gets halted. So the TUS protein stands for terminus utilizing substances, will form a complex with the TER sequences. Thereby, this TUS-TER complex will be helpful in the termination of replication in case of prokaryotes. So it won't allow the DNA B, that means the helicase, to pass through, and hence there won't be any unwinding of DNA strand. Thereby, the replication gets halted. The TUS-TER complex is essential for the termination of DNA replication as far as prokaryotes are concerned. And it is highly sequence specific,